Hey biology students, in this video we're going to talk about mutations. So now the cartoon on this slide applies to our lecture where we have these DNA molecules sitting around the campfire and one of his bases were gone. So what happened? Well, this is the case of mutation where we see a change in the DNA molecule. Now the concept of mutation most of the time has a negative connotation, as though something harmful is going to occur as a result of a mutation. And that's definitely possible. But in this lecture, I want to talk about what mutations are and that mutations are not always harmful. Sometimes they have no effect, and also they can have a positive effect. So let's start actually by looking at a mutation that has a harmful effect. And this is the case of certain genetic diseases. So this is a story of a family that has their first child, and their first child is named Hayden. And Hayden was born as a normal child, but begins to develop some unusual signs where he seems to be developmentally delayed. And I'm going to take you into their story a little bit. We're going to pick up to about here where they learn what is going on with Hayden and that he does actually have a genetic disease. Let's take a look.
It turned out that Hayden had Tay-Sachs disease, a genetic condition that slowly destroys a baby's brain.
What happens is the child appears normal at birth and over the course of the first year begins to miss developmental milestones. So at 6 months, a child should be turning over. A child is unable to turn over, to sit up, to stand, to walk, to talk.
Tay-Sachs begins at one infinitesimal spot on the DNA ladder when just one letter goes wrong. Say this cluster of atoms is a picture of that letter. A mistake here can come down to just 4 atoms, that's it. But since genes create proteins, that error creates a problem in this protein, which is supposed to dissolve fat in the brain. So now the protein doesn't work, so fat builds up, swells the brain, and eventually strangles and crushes critical brain cells. And all of this is a result of one bad letter in that baby's DNA.
That's my boy.
Tay-Sachs is a relentlessly progressive disease. In the years since his diagnosis, Hayden has gone blind. He can't eat solid food. It's harder and harder for him to swallow. He can't move on his own at all. And he has seizures as often as 10 times a day.
For children with classical Tay-Sachs disease, there's only one outcome, and children die by age 5 to 7, sometimes even before age 5.
Okay, so that is the story of Hayden, who has the genetic disease called Tay-Sachs disease. And you can see in this picture, this is a lysosomal storage disorder, which means that there's a mutation in his DNA that's inherited. So he's born with it. And that mutation is on chromosome 15. And in the previous lecture video, we were describing the very important concept, so important that it's called the central dogma in biology. And recall, the central dogma in biology is the flow of genetic information. So the flow of information, DNA is remember the cookbook of the cell holds all the recipes. And so here we're talking about only one specific gene or recipe. And this is the gene that codes for an enzyme, a protein. So this DNA, the central dogma, is that the DNA is copied into mRNA, And then that mRNA is used to build the protein. And the protein we're describing in this genetic disease is called beta-hexokinase A. And that's an enzyme. How do we know? That's right, the ending, the ACE. So this is an enzyme. It's a normal enzyme that we all have. And in this picture, it's showing you it actually is an enzyme in your lysosome organelle, one of the organelles inside of our cells, that helps in the digestion or breakdown of a certain type of fat. So this is the GM2 ganglioside. So this is a type of fat, and it helps, this enzyme breaks down that fat so it doesn't build up. Well, what happens is if you have Tay-Sachs disease, what happens is the mutation that occurs here in the chromosome does something to that DNA, alters that DNA. And if the DNA is altered, notice the flow of information in the central dogma. So the DNA is altered, it's going to alter mRNA, it's going to alter your protein. So in the diagram, they're showing us that we have the mutation that's now affecting the enzyme. So we're going to write mutated enzyme. And if it's a mutated enzyme, will it bind to its substrate, the GM2 ganglioside fats? No, it won't be able to bind to it because the shape of proteins is so very critical to their function. That is a really important theme in biology that I hope you have recognized this semester. Shape matters when it comes to the proteins, especially enzymes, because enzymes bind so specifically to a particular substrate molecule. And if they can't fit like a puzzle piece with their substrate molecule, then it will not function. So the enzyme is no longer functioning. And what happens is that the fats build up in specialized cells called neurons in the brain. So this is actually a neuron cell. And they build up to toxic levels that actually destroy the brain cells. And that is why Hayden has those unbelievably horrible symptoms. I have seen this video many times, let's just be honest, but every time I see that video, my heart goes out to that family. I can't imagine what that must be like to basically watch your baby deteriorate in front of your eyes, where he goes blind, he can no longer take solid food, he's having seizures multiple times a day as those brain cells are just destroying progressively. That is heartbreaking. And I like to give you a little glimpse into that world. Most people probably are unaware of genetic disease. You may know people that have genetic disease, but it's not necessarily everybody's reality, but it's definitely that family's reality. So this is an example of when a mutation can have a harmful effect, can actually have a disease effect. And you notice also in the story, they tell us it's only one DNA letter, or what we call one base that's affected, that causes this. That's pretty astounding, actually. Let's take a look closely at how that could happen. This is a practice problem. It's based on the previous lecture video that you watched, where we learned about the central dogma. And the central dogma, recall, is that the DNA is first copied in the nucleus into a messenger RNA sequence by a specialized enzyme called RNA polymerase. And then that messenger RNA leaves the nucleus where it enters the cytoplasm and binds to ribosomes, where the tRNAs transfer in the corresponding amino acids based on the sequence of the mRNA. use the chart here called the genetic code and see if you can self-test yourself here. Can you transcribe from the gene sequence, and can you translate into the amino acid sequence? So I'd recommend you try that right now in your notes and pause the video. So I'm now going to show you the answer. So in this practice problem, you were given the strand of DNA that we call the template strand. So you remember one of the strands is called the template strand and the other is called the coding strand. If you are given the template strand, you know that because the 3' is on the left and the 5' is on the right. And when you're given the template strand, it means you produce a complementary mRNA sequence. So you apply the base pair rules, with the only difference being that there is no thiamine base in RNA, so you replace thiamine with uracil. So notice we had to do that here as we wrote the complementary sequence, the mRNA sequence, we had to put in the U's here instead of T's. Then how do we get the amino acid sequence? You get that from using this chart here that we call the genetic code. So recall, you need to learn how to use this chart. You just don't memorize it. Okay, so we look for each 3 letters of the mRNA. What do we call that? Do you remember? Every 3? Let's circle them. What do we call that? That's right, we call them codons. So here we have 5 codons in the mRNA. Therefore, we're going to have 5 amino acids in our protein. So the first one we look for is GGG, glycine. That's why I've written in GLY, which is the 3-letter abbreviation for the amino acid called lysine. So you follow that through looking for each codon, each of the 5, and plugging in from that the corresponding amino acid. So that's how you decode the mRNA sequence to determine the amino acid sequence of the protein. So that's a review. Now, in this lecture video, we're discussing mutation. So I'm going to go to the next slide. I'm going to change colors. Let's do a different color like green. And now I'm going to say there's a mutation in the DNA. So there's going to be a mutation in the DNA, and this A here, we're going to cross it out, and we're going to put a G there. Okay. So now the sequence would be a GTA here. That is a type of mutation. So when we have a change in the DNA sequence, that is called a mutation. So mutations are always derived in the DNA, a change in the DNA sequence. The specific type of mutation that I've modeled here, where we get a base change, you see that? We had an adenine here. Now we're going to change it to a guanine there. That type of mutation is actually called a base substitution. Recall, we oftentimes refer to these letters here as bases. And so we have a base substitution if we have a change from an A to a G. That would definitely be a base substitution. It's a single base change. And that's exactly what causes Tay-Sachs disease. It's one letter. One letter is changed in a very, very important gene. So now let's talk about how that's going to affect the protein, the hexaminidase A enzyme that's built from this recipe, this gene sequence. Okay, so now we're gonna, I left the originals, what it originally would transcribe and translate to, but let's go ahead and do now the mutation in green. So in mutation, the first three are the same, right? But now we have a G in this spot. That means there's going to be a C in the RNA, and then we'll continue here. And the rest of it will be fine, right? So we'll continue. That was the only area where there was a change. So in this example, we ended up with this codon changing. So the original codon was UAU, and now it's CAU. So it originally coded for tyrosine as the amino acid, but now it's CAU. So what's CAU? Well, let's take a look. CAU on the codon chart codes for an amino acid called histidine, which is shortened as HIS. HIS. So now we have a different sequence. So we have a histidine here. And yeah, everything else is still the same, except for that one amino acid that's different. This is really where it gets sort of impressive in a way here, that it's a single spelling change in the DNA. Just one letter is changed, but it happens to be in a very important gene sequence. It happens to make enough of a difference that now, because of this histidine in place of the original tyrosine, the chemistry of this specific amino acid is going to interact in a different way in the folding of that protein. Because really, this is just showing us primary sequence. This is just the order of the amino acids. That doesn't really tell you about the shape. Remember, this is going to dictate the shape of an enzyme, because enzymes are three-dimensional proteins. So that enzyme needs to have a specific shape to it, like that. But if we get a mutation in the DNA, do you now see how that mutation in the DNA gets passed to what codon is put into the mRNA, and then that gets passed to what amino acid is added in the building of this particular protein? So that mutation, in some cases, can have a very significant effect, altering this active site of the enzyme. So if the substrate molecule, let's say, looks like this, a rectangle, okay, that's our substrate, and it needs to fit into the active site, well, that substrate is not going to bind correctly. We're not going to get interaction there. And we're going to get, we're not going to get that fat degraded in the brain. We're going to get Tay-Sachs disease. Now, I have to be honest, I simplified this, and I believe it actually is a different sequence than the one I actually put in here, but I just did this for showing you the effect, that a single base change can actually affect the sequence. The reality is it's a much longer sequence, but these changes can have significant effects on the protein. So this is an example where a mutation can be harmful. Let's back up a minute and try another mutation. I'm going to induce another mutation. Okay, now I'm going to take this adenine here, and I'm going to replace it with a guanine. Different spot, I know. And then on the previous one, I altered this first A. Now I'm going to alter this A into a guanine because I want to show you something else. So now we're gonna transcribe the gene, and now we're gonna put a C. We're gonna put a C in there. So it used to be UAU, and now it's UAC, and the rest of it is the same. Okay. So the original codon coded for tyrosine. What does UAC code for? Oh, look, hey. Both UAC and UAU code for tyrosine. Look at that. Remember, that's a feature of the genetic code, and we call that redundancy. Redundancy means when there's something that does the same thing. So we can have multiple codons where even a mutation in one of the letters is all off, it still codes for tyrosine. So we would still, in the amino acid sequence, we would still have tyrosine. So this is a situation that can occur in the cell where a base substitution is induced or occurs spontaneously, and that has no effect. So we would say in this situation, this would have no effect on the protein. So be careful about that. Sometimes I will definitely give you questions like this on the next exam, where I give you a codon chart and ask you to transcribe the gene and translate to the protein. And I will give you a mutation and I'll ask you, is this going to affect the protein structure? And if it codes, the mutation results in the same amino acid being added, then the answer would be no effect. So there is the possibility, because of the redundancy of the genetic code, that a mutation will not have an effect on the protein structure. Or we can be like this situation where it just happened to be the other adenine got mutated that did have an effect on the protein structure. This case, a harmful effect. Let's talk about another genetic disease. This is called sickle-cell anemia. This one we see exactly where the mutation occurs. So this is the original DNA in a normal situation. This is the RNA that is transcribed from the gene. And then this is the amino acid, glutamic acid or glutamate, normal protein. This is actually a protein called hemoglobin. So this affects a protein called hemoglobin, and the hemoglobin is found in your red blood cells. So it's in your red blood cells. And it also affects the shape of your blood cells. So in normal blood, your red blood cells are round. Like pictured up here, they're showing us normal red blood cells, nice and round with lots of hemoglobin. And hemoglobin is actually what binds to oxygen and transports oxygen throughout your body. It's a very important protein. So what about the disease? What happens? A mutation in a single base pair. That again would be an example of a base substitution. That affects a single letter, and that also changes the amino acid. So in this case, this is not a case where there's redundancy that saves this. Instead, this actually changes the amino acid. And because there's a mutation in the hemoglobin, so hemoglobin becomes mutated, here's what happens to the red blood cells. The red blood cells become what we call sickle shape. This is the red blood cell. You can see it here. And what happens is they look sort of like bananas under the microscope instead of being round. Now, this actually causes a disease because these sickled blood cells cannot transport oxygen as effectively as the round red blood cells. But I want to show you something. This is a map showing you where the sickle cell trait or the sickle cell allele is most commonly found. And if you recognize the map, this is Africa. So it's showing us we're seeing higher percentages in Africa of people that have this particular mutation in these regions. And if you overlay that with this map, this is showing us regions where malaria is endemic. And that word endemic just means common in the population. So there's mosquitoes that live in Africa that carry a parasite called falciparum. And those malaria zones overlap with where the sickle cell allele is found in those people. Why is that? Well, researchers went in there to try to understand that. And here's the cool part. they learned that if you carry the sickle cell allele, and if you're heterozygous, which means that you carry one copy of the normal allele and one copy of the mutated allele, then only 50% of your blood cells are sickled rather than all of your blood cells being sickled. But if you have at least some of your blood cells sickled, guess what happens? The parasite is unable to invade the blood. So the malaria parasite cannot infect your blood. So you get protection from malaria. Isn't that amazing? So this mutation that causes a shape change in the red blood cells protects you from invasion by the malaria parasite, where if you have normal red blood, the normal blood cells actually get invaded by malaria. But people who have at least a majority of their blood cells that are sickled, they'll be protected from it. And if you carry both chromosomes with this mutation, then you're fully protected from malaria. So this is an example of where a mutation causes a change in a protein, okay, causes a change to hemoglobin, but it actually benefits your survival. It provides a survival advantage if you live in an area like here in Africa, West Africa, where malaria is common. This is actually an evolutionary principle. It's called selection. We'll talk more about it in a future episode, but I wanted to show you that mutations can be beneficial. If you lived in if you live in Africa where malaria is very common, you would want to carry this allele because this would be a protective part of your blood to keep this parasite from causing malaria and from killing you, because malaria kills millions, hundreds of millions of people every year. And this is a protective mutation. So this is an evolutionary concept because many of these changes in our DNA actually create new proteins that have a protective advantage. It's not always going to be a harmful situation. So we've learned in this video that mutations are changes to the nucleotide sequence of DNA. And if you understand the central dogma, you understand that DNA is used to create proteins. So those proteins do get affected by the DNA sequence. We learned that mutations can be harmful, like an example of Tay-Sachs disease. They can have a neutral effect as a result of redundancy in the code, where the same codon codes for the same amino acid, or can be beneficial. The example being like sickle-cell disease, where if you live in Africa, there's an advantage because the mutation protects you from malaria. What causes mutations? We really put this into two categories. So mutations can be and are natural, spontaneous. In other words, they're a product of natural biological processes within your cell. In a previous video, we learned about DNA replication. We learned about this important enzyme, DNA polymerase. But I want to tell you here that DNA polymerase, when it's copying your DNA code, it actually makes spelling errors. Have you ever made a spelling error before? Well, it's built into your biology for there to be spelling errors in the copying of your DNA code. That is a normal thing. And that is an important thing to the survival of species, that not all species are clones, identical genetic clones of each other. Because if they were, well, we'd all be wiped out, wouldn't we, by a single infectious disease. But there's variation. It's the same reason why we see some people surviving COVID-19 and some people not. It's because there's variation in their code. Some of them have a protective immune response and some of them do not. And that's built into your DNA. And so in the copying of your DNA, there are little spelling changes that occur. There's also a natural process called recombination that occurs in meiosis. We learned about this in the last unit. crossing over between the homologous chromosomes. It creates new alleles, or it can. So these types of mutations, if they occur in germline cells, they can affect future generations. So these mutations, like with genetic diseases like Tay-Sachs disease and sickle-cell disease, that's something that does get passed on to future offspring because it ends up in the gamete cells. Induced mutations are not usually passed on in the gamete cells. So induced means something you're exposed to as a child or as an adult. Chemical mutagens, physical mutagens, and pathogenic mutagens all fall into this category. So you're exposed to cigarette smoke. Well, large quantities of cigarette smoke has been known to damage DNA, and by the way, damaged DNA can lead to cancer. So there's a very direct link there between cigarette smoke and increased risk of cancerous cells in your lungs developing. Same thing with radiation. So you probably know exposing yourself to large quantities of sunlight radiation, UV radiation, can lead to skin cancer. Again, because we know that's a physical mutagen to cells in their DNA. There's also pathogens, so certain viruses and bacteria can also damage your DNA. A good example of that is HPV, which causes cervical cancer. But an important thing to note, they don't usually pass on to the children because usually this is damage that you've done in your somatic cells, like your skin cells. So if you get a sunburn and you end up getting skin cancer, well, you have mutations in your skin cells, but those are not in your gamete cells, so those wouldn't pass on to future generations. Anyway, so that is the story of mutation, a very, very important theme in biology, especially very important to understand when it comes to how does evolution work, which we'll describe later in a future video. Talk to you later. Bye.